Hey, what up all my tooth doctors and doctresses? Welcome to another video at the Tooth Factory. Today we're going to continue the Principles of Pharmacology Part 2 with Pharmacodynamics, created by Dr. Kanan Shah and presented by me, Dr. Rishi Shah. Today's lecture will comprise of the principle summary we looked at in the Pharmacokinetics lecture as well, the drug receptor, dose responses, and intrinsic activity. These three topics from pharmacodynamics are what we're going to talk about one after the other, and we will end our lecture today with important terminologies. So here's the difference between pharmacokinetics and pharmacodynamics. Remember that kinetics means to move the energy of movement. That means what the drug goes through when it passes through our body. In other words, pharmacokinetics is what the body will do to the drug while it passes through our body, while it moves through our body. Pharmacodynamics, on the other hand, is what the drug will do to the body itself. So how I remember it is between the two terminologies, pharmacodynamics has a D, and therefore drug comes first, what the drug does to the body. That's how I remember it. Kinetic means that it moves. So when the drug moves through our body, how does the body deal with it? That's how I remember the two differences. Today, our topics are divided into drug receptors. Basically, we're going to talk about major receptors and how they act with the drug. Then dose responses with specific concepts of affinity, potency, and efficacy. Very, very important to focus on, guys. So please have your notes out. We will talk about this in a conceptual manner. So when the questions are asked in the exam, such as the AFK or INBDE, we know how to approach them. And then we will end our lecture today with intrinsic activity. By that, what we mean is agonist and antagonist properties of drugs. Okay, so first things first, receptors. Now, we need to understand that unoccupied receptors does not influence intracellular processes, which means this receptor can be sitting here all alone, right, without any drug acting on it. Until that drug occupies that receptor, there will be no effect intracellularly. Nothing. Zero. However, receptor with bound agonist is activated. It has altered physical and chemical properties of the intracellular nature, which leads to interaction with the cellular molecules to cause a biologic response. So very simple, right? We got receptor. Then we got a drug attaching to the receptor. Then we got agonistic properties, which means to cause an effect. And it leads to biological response. Very simple, right? So here it is, the drug goes to the receptor. Receptor is now activated with the yellow halo there, and a signal is transducted within the cellular molecules, and biologic response is produced. Many times it's the desired response of the drug, sometimes it's the side effects. However, a biologic response, good or bad, is given out. Okay, so let's take a look at various drug receptors. There are a total of 4. This slide has 2 and the next has the other 2. So number 1, ligand-gated ion channels. What does that mean? Well, drugs will open or block ion channels. The intracellular effects are produced by manipulating the membrane potential of the cell, which means the drug will come to these receptor channels and change the charge on it. The membrane potential of the cell will change, and therefore the ion concentration inside the cell will also change. And when that happens, we have a biologic response. These ligand-gated ion channels are represented by the example of cholinergic nicotinic receptors. So this is our next lecture. We will talk about the CNS and ANS. This is where we talk about cholinergic and nicotinic receptors in the cholinergic cycles. The next type of receptor is G-protein coupled receptors. These are very important receptors, because what happens in G-protein coupled is upon drug attachment to the receptor, a cyclic AMP, which is a second messenger, is produced, which then conducts protein phosphorylation or dephosphorylation. Now, this process is responsible for causing the intracellular effects, the biological response. So this now becomes a three-step process, right? Cyclic AMP, then phosphorylation of the proteins, and then the biologic response, intracellular effects. Examples are alpha and beta adrenergic receptors. We will also talk about these later on in the future lectures, and this is from the adrenergic system. The third one is receptors as enzymes. Now this one's kind of cool, guys. So drugs upon attachment to the receptors will produce regulatory biochemical reaction by phosphorylating proteins, such as surface protein kinases. Now, the important point from the whole sentence here is regulatory biochemical reaction. See, such proteins are located on the cell surfaces. Basically, when the drug will act on the receptors, these are technically literal surface proteins that then carry forward a particular action of regulatory in nature. Regulatory. For example, insulin. And last but not the least, the intracellular receptors. Easiest to understand. See, it's only lipid-soluble and non-ionized drugs that can cross phospholipid bilayer to enter the cell. Remember this concept, we repeatedly talked about this in the pharmacokinetics lecture. Once the drug is inside, the receptors are located in the nucleoid. In the nucleoid, within the DNA area, it presents alterations of gene expressions in the DNA itself. So down to the molecular changes. The nuclear matter is then altered intracellularly by the drug itself, and examples are we got steroids with soluble DNA transcription factors. So yes, basically steroids are the drugs that go into the nucleoid and affect the DNA directly. So let's take a look at what a receptor and a drug interaction could look like. So first of all, an unoccupied receptor does not interact with the protein, right? There's the drug in the extracellular space or a hormone or neurotransmitter. The receptor is unoccupied and there's no activity here. There is inactive state on the cell membrane. But once the receptor is occupied and then it changes its shape, right? The drug attaches itself, the neurotransmitter hormone attaches itself to the receptor, it changes shape and affects this alpha subunit of this GS protein on the cell membrane. That leads to the exchange of these molecules, the GDP and the GTP. So GTP takes place. However, we still have inactivity, not there yet. Once the alpha subunit of the GS protein right here, it detaches itself and goes to the adenylyl cyclase and causes an activity, that's when they're active. However, once the activity is done, the drug here is now completely used up. There is no more drug or hormone. The receptor sets back to its normal position. The shape goes back to normal. However, the alpha subunit is also dissolved, right? It's all done. It's called hydrolysis of the alpha subunit. This becomes inactivated again. It's very simple how a drug can enter its effects into the cell. Just a little diagram. Okay, so let's take a look at the drug receptors visually. This summary will help us understand all receptors easily. So number one was ligand-gated ion channels, right? Very simple. A drug or a hormone or a neurotransmitter attaches to the receptor on the cell membrane. That leads to changes in membrane potential, so changes in the membrane right here, and then ionic concentration within the cells as well. That further leads to intracellular effects. Very simple. Cholinergic nicotinic receptors are ligand-gated. This is the G protein couple receptors we just talked about. So now we understand why the drug attaches to the receptor. The receptor then touches the alpha or the beta or the gamma subunits, which then lead to activity on the adenyl cyclase that's sitting here, leading to a process called as protein phosphorylation, and of course then the biological response. So G protein couple receptors are very important in our adrenergic cycles, adrenergic systems with alpha and beta receptors. We will come back to these in the future lectures. That's why we looked at it in the previous slide. Third ones were enzyme-linked receptors. Remember the surface proteins. So this receptor here has insulin, for example, sitting on insulin receptors, the surface proteins, right? That causes an interaction within the cell. This is protein and receptor phosphorylation. That causes intracellular effects. Remember, these intracellular effects were known as regulatory biologic responses. Regulatory. Insulin causes regulatory effects. And last but not the least, the intracellular responses, such as steroid receptors. So the steroid doesn't have a receptor outside or on the cell membrane. It goes directly through the cell membrane because it is lipid soluble, and as well as it is non-ionized. Remember, these two words are very important. For anything to cross the cell membranes, we have to be lipid soluble and non-ionized at the same time. Now, intracellularly in the nucleus, there is a receptor waiting for the steroid. It will cause protein phosphorylation and alter gene expression as its process for causing biologic response. So I hope it's clear now about the receptors. Moving on to the next concept of drug responses. So once it has attached, the biologic responses occur. What really takes place within the cell? So first of all, selectivity. Now, it's very important to understand that drugs that have increased specificity for a particular receptor of choice is known as selectivity. So for example, if this is receptor A, right? This is receptor A and this is drug A. Now, it is very important to know that this attaching to this will cause a good effect. But if this A right here sat in a different receptor, then we could have side effects. So for this, selectivity is extremely important. A drug will need to be selective for the receptor. This helps to reduce side effects and produce desired effects instead. Affinity, however, is how tight the bond is. Affinity is the binding tightness or binding power of a drug to its receptor. This balances the interaction between agonist and antagonist. So, for example, if the drug A, same example here, is extremely tightly bound, right? Extremely tightly bound. Can an antagonist, which is an opposite function, basically it doesn't cause any effect, when it attaches itself to the receptor, can it remove this agonist? Well, if the drug has a very high affinity, then the receptor and that drug are inseparable. They cannot be separated. However, if the affinity is low, then that drug will detach itself from the receptors, right? We are then going to see a detachment here, and then an antagonist is going to sit here. Now, if an antagonist sits here, then we have no action. Agonist is useless. So to maintain the balance between agonist and antagonist, a drug must have a very high affinity. So we want high affinity and we want a high selectivity. Okay, moving on to the next concept, efficacy. One of the most important concepts, guys. It is a property of the agonist. Agonist means something that causes effect, right? What's efficacy? It's the number of receptors that must be activated to produce a maximum or a full response. So the two very important words, full response, right here. So when efficacy is involved, we're talking about full response. But how many receptors need to be activated? Well, if full response needs 5 receptors activated, then A, B, C, D, and E all have to have drugs attached to it. Correct? Now we have complete efficacy. However, if by any chance one of these don't get activated and gets occupied by antagonist, then we only have 4 out of 5 receptors activated. Will we have full response? Well, no, we won't have full response, and therefore we will now have low efficacy. Fairly simple, guys? Okay, so efficacy is number of receptors that must be activated to produce a maximum full response. And Emax is the maximum response of a drug at a certain number of receptors activated. So every drug will have a separate efficacy. Some drugs will need 5 receptors. Some drugs will only need 4 and still have full response. So, for example, drug A is more potent than drug B, but both show the same efficacy. So I'll leave the potency out of the equation for just a second. It's in the next slide. We'll talk about it shortly. The two drugs, drug A, drug B, both have same efficacy. It means they both are able to reach a full response, right? That's what it means. However, drug C shows lower potency and a lower efficacy than drug A and drug B, which means they only shows 50% of the full response. This is low efficacy. So efficacy means the ability to produce a maximum response by attaching to the maximum number of receptors. That's efficacy. This is about potency. So the potency of a drug can be compared using EC50. The smaller the EC50, the more potent the drug it is. So now let's talk about this in the next slide, okay? Potency. What is potency? Another property of agonists is the concentration associated property. Dose and concentration required to produce desired effects. Basically, do you need 5 grams of a drug to produce a maximum response? Do you need a 10 gram of the drug to produce the maximum same response? That means this drug is more potent than this because they both have the same response, right? If a drug provides desired effects at a lower concentration, right here, then it is highly potent. If these two have the same effects, but 5 grams for drug A and 10 grams for drug B, then drug A is more potent. And EC50 is the dose at which the therapeutic effects are produced. Remember in our therapeutic index in our previous lecture. Let's get back to the chart. Drug A is more potent than drug B. Oh, okay, let's see how. Well, drug A was able to produce a maximum efficacy, maximum response in only this much concentration of the drug provided. However, drug B also produces the full response, maximum effects. However, it needed this much concentration to do so, which means drug A becomes more potent than drug B. Now, drug C shows lower potency than drug A and B. Yes, absolutely. It just needs even more of a concentration of the drug to produce the maximum response that it's eligible to produce. So now C has low potency and low efficacy, both. So I hope the two concepts are making sense, guys. And if there's any questions to clarify this, please leave us some comments and we will be sure to help you out. Now in the second picture here, the potency of a drug can be compared using the EC50, which is where the dosage and the concentration of the therapeutic effects. The smaller the EC, the more potent the drug, which means less the amount of drugs needed and more the effects produced makes the drug more potent. Now, last but not the least, the intrinsic activity. We're going to talk about the agonistic properties and the antagonistic properties to summarize a whole lecture. So first of all, what is intrinsic activity? It's a form of drug behavior, right? It's how the drug behaves. So it's the ability of a drug to produce a biologic response after attaching to the receptor. That's it. It's the ability to produce a biologic response. it's got the all or none law. It can either be agonistic, which is all, or antagonistic, which is none effects. Now, keep this in mind, guys, that agonist means the biologic effect is produced, and antagonist means that it does not produce opposite action. However, the biologic action is prevented. This has no opposite action. It just has no action at all, right? It prevents the agonist from acting at the receptor by occupying the current receptor. So agonists have full agonists and partial agonists. Antagonistics are divided into competitive, non-competitive, and physiologic. So let's take a look at each on a singular basis. Agonists. We got biologic effect being produced. A full agonist and a partial agonist. All of them have the same types of patterns. See, full agonists have a high affinity, very tight bond, right? High efficacy means maximum numbers of receptors activated to produce the maximum response, and high intrinsic activity, which means all law from the all or none law. Basically, the full agonist will mimic the biologic endogenous regulatory effects, such as endorphins, which have all receptors everywhere. Every receptor, whether it is CNS, whether it's in the ANS, whether it's on a target organ, everywhere a full agonist will check the boxes and cause the maximum effects, leading to a positive biologic response with a very tight bond that is immovable. However, a partial agonist has a low affinity, not as tight of a bond. It can be removed by the antagonistics very easily. And a low efficacy. It doesn't produce maximum response. And a low intrinsic activity, in other words, it doesn't always have the all or none effect. So basically, it produces partial response, right? Partial response only, and does not cross the blood-brain barrier to elicit effects fully. That drug will ignore the receptors in the brain. So for example, if we avoid the CNS from this equation, it only activates certain other areas. That means not everything got, activated. The brain was left out of the equation. Then we call it partial agonist. This is because it could not cross blood-brain barrier. Moving on, a full agonist produces complete activation of a drug receptor at high drug concentration. So for example, the red line here is of a full agonist, right? A receptor activity of 100% is reached at the highest concentration possible. That's a full agonist. Every single receptor is done and dusted. We got a high efficacy, high affinity, a high intrinsic activity. Everything is perfect. However, in a partial agonist binding results in less than 100% activation, even at high concentration. So no matter how many milligrams or how many grams of drugs you give them, what the concentration is, the partial agonist will never have a high efficacy or high affinity or a high intrinsic activity. However, it will only have low of each category. It will, for example, ignore the CNS or blood-brain barrier, right? It will not cross that. See, for example, in the inverse agonist, we didn't talk about this in the previous slide. However, it's very interesting that inverse agonist will produce a response below the baseline response measured in the absence of drugs. This agonist will not produce the opposite action, but it'll produce an effect. However, it'll be still lower than the baseline effects that happens without the drug in our body anyways. So in this example, approximately 12% of the receptors show constitutive activity in the absence of agonist. Right here. A baseline is 12% receptor activity, right? 12% receptor activity right here. However, a full agonist will bring that to 100%. A partial agonist will bring that to about 50-60%. So I'm going to put 60%. and an inverse agonist will bring that all the way down to 0% below the baseline. So just as a summary. Very important diagram, by the way. Okay, we're now on to antagonists. They do not produce opposite actions. Their job is not to produce opposite of agonists. Their job is to prevent the agonistic activity. Basically, their job is to stop the agonist from attaching to the receptors and causing biological response. They do have high affinity. Well, you have to. If you want to prevent somebody from entering your house, then you have to have a very, very tight lock, right? And that lock is antagonist. It produces no intrinsic activity, which means no thief will be able to steal from within the house. Just a little way to remember that. A competitive antagonist, a non-competitive, and a physiologic antagonist are the three categories. So let's take a look at them one by one. A competitive antagonist, such as acetylcholine and atropine in the cholinergic systems, and the morphine and naloxone in the opioids. One over the other are reversible in action on the same site of receptors. However, they work to decrease the potency of the agonist. The dose of the agonist must increase. What does this mean? That they both, like for example, ACH, the acetylcholine, and the atropine, both affect cholinergic receptors. They both affect cholinergic receptors. However, they are reversible on one another. They work to decrease the potency of one another. And therefore, just to overpower that, we're just going to increase the potency, which means we're going to increase the concentration of the agonist again. So for example, if ACH is sitting on that receptor, it will not let atropine work. Now, if we increase the dose of atropine, ACH will fall out of the cholinergic receptor and sit on the cholinergic receptor and do its job. That's what reversible means. Non-competitive, however. Non-competitive means I'm not here to compete. I'm just going to do it once and that's it. I'm irreversible. There's no competition. There's no question, right? Basically, they act on different sites on the same receptor. And the work is done to decrease the efficacy of the agonist. Now, this is a little different. We got one receptor. We got two sites. The agonist will sit here and the antagonist will sit here. This drug right here will work to reduce the efficacy, which means it will prevent a maximum response of the agonist irreversibly. It doesn't care. There is no competition. I have my own place here. I'm going to sit here and not let you act at all. Therefore, the efficacy is compromised. So remember the two differences. Competitive antagonist, non-competitive antagonist. Competitive means the two opposite acting drugs can, fight for each other on the same side of the receptor though. That has to do with concentration of one over the other or the other over the one. That's all. It's very simple. However, in non-competitive antagonists such as organophosphates, also known as sarin gas, they permanently inhibit respiration, right? For example, in historic references, there was a sarin gas attack in Syria years ago where the permanent inhibition of respiration took place and people could not breathe because that organophosphate sat on the receptor and said, that's it, I'm going to prevent the agonistic activity of breathing completely and irreversibly. There is not going to be a maximum response of breathing, no efficacy, done and dusted. Dose of agonist is irrelevant. Then even if you keep giving them oxygen, it doesn't do anything because the antagonist just sits there and prevents agonist. Very dangerous, you guys. And last but not the least, the physiologic antagonist. This is a little different because this is opposite action causing. I know we said it does not produce opposite action. Yes, on normal, general basis, antagonists are not designed to do so, but there are some physiologic antagonists, such as epinephrine and its opposite physiologic antagonist of histamine. They both are actually agonists, right? They're not antagonists. They're both agonists. They're two different agonists that bind to two different receptors itself, both receptors. and produce opposite biological response. Now it all matters is which one is more. Is epi more or is histamine more? And that effect will work. For example, vasoconstriction at alpha adrenergic receptors and vasodilation at histamine receptors. So epinephrine causes vasoconstriction while histamine causes vasodilation. Opposite actions, but they're not antagonists. They're two agonists with physiologic antagonistic properties. So just to clarify those words precisely, guys. We'd already looked at this diagram here, but in this sense, the drug with non-competitive antagonists, for example, this one in orange line, will prevent efficacy. Remember, maximum response was right here. It cuts its response into fractions. However, a competitive antagonist allows a full production of response, but it needs a higher drug concentration because, it sits reversibly and says, I'm not going to let you work until you bring more of your army. So, EC50 of a drug in presence of a competitive antagonist is all the way on the right side of the chart because we just need so much more drug concentration to produce the effects necessary in presence of a competitive antagonist. However, the EC50 for a drug alone or in presence of non-competitive antagonists is the same. Very similar, actually. The only thing it prevents is maximum response. So remember this. A non-competitive will decrease efficacy. A competitive will decrease potency. A physiologic antagonist, right? A physiologic antagonist will have two agonists with opposite actions on different receptors. These are the antagonists of the intrinsic activity. Okay, so let's end the lecture with some important terminologies. What is synergistic response? What is synergism? Synergism is when using two drugs to produce more than double effects occurs. So 1 + 1 = 3. Basically, when you have drug A and drug B given, it gives you more effects than drug A and B combined. So even more effects than that. Additive effect is using two drugs to produce no more than double. Basically, you just want A plus B equals, well, A plus B. And adverse effects is unintended effects that are produced within therapeutic doses. We call these side effects, such as nausea, vomiting. Right? These are unintended. You don't intend to give these. However, within therapeutic doses, they occur. Toxicity, on the other hand, are very similar. They're unintended effects that are produced. However, this is in excess of therapeutic doses. This is overdose effects, not side effects. And last but not the least, idiosyncratic reactions. These are unpredictable reactions to a drug specific to an individual, only not all, right? For example, you're looking at me being allergic to a drug with skin rashes excessively. However, if you were to take that drug, you will not have any effects. So that is an idiosyncratic response on my end. Some people may have a genetic predisposition identified. Now, I say some, but the research suggests that idiosyncratic responses are genetically based responses. And effects are displayed at first exposure itself. So when you have to answer a question that says, is this an idiosyncratic reaction or an adverse reaction, please look for genetic predisposition and also look for unpredictable reaction, which is individual specific on the first response, first exposure itself. Okay, guys. So the resources for the lecture are pasted below in the description box. We did Lippincott and Little and Fallis for our pharmacology as always. For more academic lectures like this one, please stay tuned at the Tooth Factory and we will be happy to help you. Please don't forget to like, share, and subscribe. It really helps us grow, you guys. and it helps us bring you some beautiful lectures like this one. Thank you very much. Have a great day.